lot many students, and as the head of the department has already said that there are many students in the audience, so I had thought earlier that I'll make this presentation. I will start with something very simple and gradually I'll move towards the complex part, and that is what actually is the title of the talk as well. So I will be starting my presentation, and I hope that those who are aware of cytogenetics will also get a feel of what this subject has, how this subject has evolved over the years, and those who are the students who are initiating, perhaps they will find it interesting, and that is perhaps what is required, because we want a new energy to be put in so far as cytogenetics is concerned. I will tell you the reasons why. Are the slides visible?
Yes, sir, visible.
Okay, so let us start. I am talking today about cytogenetics to genomics, the journey so far. As you already know that I belong to School of Biotechnology, University of Jammu, and currently I am the Vice Chancellor of the university. So again, I would convey my gratitude to all of you for thinking that I could be delivering this lecture, and thank you for giving me this opportunity. Professor A. Abraham Endowment Lecture 2020. As has already been said, Professor Abraham was a doyen in cytogenetics and plant breeding, and he has been an institution builder. He has made several institutions. So I bow with humility in front of him, and I wish that I get that kind of blessings from him, that I would be able to justify this particular lecture. Let me first introduce University of Jammu. It was established in the year 1969. We have already crossed 50 years of existence. It's a NAC accredited A+ university. We have NIRF 52nd rank, and it's a category 1 university declared by the MHRD. It's a multi-faculty affiliating university, and we have 7 off-site campuses. This is our iconic building of General Zorawar Singh Auditorium, and these are the few pictures of the campus. This is the School of Biotechnology, where we started our work post 1999. So School of Biotechnology at the moment offers 3 MSc, MPhil and PhD programs. Biotechnology is our main program. We also have biochemistry and microbiology, and we also run a PG diploma in bioinformatics. The department has been ranked by the evaluation by DPT, the Government of India, and it is in first place of the country. It gives me a lot of satisfaction and pride to say this. And MSc Biotechnology program funding has been now continued for next 5 years by the DPT. Let us come to the talk, cytogenetics to genomics, the journey so far. I'll start somewhere very basic as to how it started. About 120 years ago, Bovary, it is in Germany, and Sutton, USA. So it's a very famous combination, Sutton and Bovary. The cytologists, they fully established the chromosomal basis of Mendelian inheritance and the chromosome heredity. They established the chromosomal basis. Chromosome, the catalyst was later called as studied the site meiosis 1949. This is called the age of classical cytogenetics. The second half, 1950 onwards, is the age of molecular cytogenetics. Of course, there are different views. Some people say there are 4 phases, but let us say that there are just 2 phases at the moment. Discovery of DNA structure by Watson and Crick in 1953. Actually, after this discovery, molecular genetics and cytogenetics, they both bloomed. Scientists began to know that DNA is organized into chromosomes. Now, it was between 1950 and 1970 that many advances in cytogenetic mapping in crop plants took place, and the major advances in cytogenetics of polyploids using wheat as a model for allopolyploid and potato as for autopolyploid inheritance. So these 2 models were taken and extensive work was done between 1950s and 1970s. Subsequently, cytology and genetics, they became 2 synonyms. So cytology and genetics synergistically contributed towards our understanding of several phenomena. There are some examples, very prominent ones. For example, cytogenetic studies in Drosophila established the fruit fly as a major genetic model. The studies by Morgan and co-workers in 1915, and then discovery of polytene chromosomes by Painter in 1933, and even today Drosophila continues to be a genetic model. Even the complete genome was sequenced in 2000. Molecular cytogenetics helped in understanding the organization of DNA in chromosomes, how chromosomes replicate, and ensure faithful transmission of genetic information. So this is possible only with the help of molecular genetics, molecular cytogenetics. Now, strangely enough, although we are tracing the between 1950 and 1970, but by 1970s, cytogenetics was thought a dead science. It was thought that there can be no further progress. Whatever has been achieved, and it's a dead science now. However, still many advances were made in the cytogenetic mapping of crop plants. As I said earlier, wheat emerged as a model in cytogenetics. In 1954, and valuable aneuploids have been mostly discovered in polyploid plants because loss of can give many ploids. But aneuploids including monosomics, ditelosomics, double ditelosomics, by and so and those talks even on today then came the revolution because of development of Gauss 1969. Now these 2 scientists used isotopes to label probe to detect the RNA sequences in cytological filtration. Now this was a very exciting discovery, and this technique, development of in situ hybridization, actually brought in the revolution in cytogenetics. So it's a new life which was given to cytogenetics. Those who thought that science, thinking that need to be done and can be done now. However, this technique, so a relief came in 1982 when Langer Schaefer and co-workers, they developed a non-radioactive, that was immunological in situ hybridization method using fluorochromes for signal detection. now named fluorescence in situ hybridization, in short FISH, and it has been used widely in human, animal and plant cytogenetics research as well as in routine clinical diagnosis. Even today, this technique is used very, very extensively. Now this FISH or fluorescence in situ hybridization has been used for physical localization of DNA sequences, when you can use repetitive DNA sequences as probes, or you can use multi-copy gene families as probes, or you can even use low or single copy genes directly on the chromosome. So you can actually localize the position of these sequences on the chromosomes, and they get wherever they are hybridized, such sequences you can watch them under the fluorescence microscope, and they will always fluoresce because they have a fluorescent tag with them. Now FISH has helped in chromosome identification and molecular karyotype construction. Besides, they have been used in with back libraries, and I will explain later how those back libraries have helped. Another variant of fluorescence in situ hybridization FISH is genomic in situ hybridization, that is FISH, and it was developed first by Finkel and co-workers in 1986. This is a special type of FISH. The difference between FISH, you use any sequence as the probe, but here in genomic in situ hybridization, you use the complete genomic DNA of a species as a probe. Now this is a very powerful technique for genome analysis. If you have a polyploid species and you want to know who are the progenitors of this polyploid species, who are the parents of this polyploid species, then you can use FISH. You can use one genome, complete one genome, you can label it with the fluorescent probe, and you will belong. This way, the others, they have been now this genomic in situ hybridization can also be used for detecting chromatin introgression during interspecific hybridization. If you develop a cross between two species, and then in the hybrid you want to know which segments of one species of a chromosome belong to one species and which segments belong to another chromosome, you can use this genomic in situ hybridization. It can also be used for identifying alien addition and substitution lines. These are important lines. So fitting as fiber is concerned, which in 1996 France and co-workers, they developed the even more powerful technique of fiber fishing. This time they used extended DNA fibers as targets. Instead of chromosomes, they used the extended DNA fibers as targets. So fiber fish greatly improved the sensitivity and resolution of fish technique and allowed the mapping of probes that are 1 kb to 1 megabase pair apart. So you can understand the power and sensitivity, the resolution which it brought to the cytogenetics. So it's a very powerful technique. The DNA fibers are made from the interphase nuclei. You have to isolate the nuclei and then make the DNA fibers on a slide. And on that slide, use the normal probes. The fish probes became very, very so far as rice genome sequencing project was concerned, because even with the conventional probes, conventional markers, they could not closure of the gaps, and that became possible for fiber fish. Now what are the toolkits of a cytogeneticist? Combining the fish technique on different DNA targets allows us to map the differentiated chromosomes at a higher resolution. The fish is that, and because of this specificity, and relative rapidity of detection, fish remains the technique of choice. Even today, more than 30 years have passed since the fish was used in plants, and it continues to make important contributions in the areas such as genomic structure and gene expression studies. Fish has also been used in meiotic pairing analysis and other genomics and molecular biology studies. Now I just wanted to briefly touch upon these two things, that you know the organization of a chromosome. The plant or even animal or human chromosome, if you see the parts of that, there is a very important component chromosome that was not there, then the chromosomes will not segregate during anaphase. Structure with which split, I just dropped that a very important component, and mapping of this centromere. Where is this centromere located? This became possible because of this fish technique, and you can see from this very slide, I am just trying to say, in yeast, mammals, Arabidopsis, there are these repeat sequences which are present in the centromere. They were used as probes in order to map the centromere in the chromosomes. Similarly, at the ends of the chromosomes, because chromosomes are linear structures, so in order to prevent them to become circular, there are structures which are known as telomeres. So telomeres are repetitive sequences which are present at the ends of the chromosome. So a typical telomere in plants is a tandemly repeated unisatellite sequence. In Arabidopsis, the sequence is TTTA GGG. This is repeated several thousand times at each end of the chromosome. Now these telomeres are very important, because in telomeres then there will be no linear structure of a chromosome, and if the chromosomes are not linear, they will not segregate properly, and there will be no there will be a lot of problems so far as meiosis is concerned. So it will be a circular chromosome whose segregation will be problematic, and the plant will be sterile. Now again, I just wanted to give you this example here of our my own research in my own research lab, which we did, which is known as Plantago ovata. This Plantago ovata is source of isabgol, and you can see that we have tried to use different sequences. For example, the ones here on the left, you can see them, these are the repetitive DNA sequences, and in the center this is typical C banding, by which you can differentiate the heterochromatic. These are the dark structures which you see here are the heterochromatic regions, and the lighter ones which are visible are the euchromatic regions. Generally, it is believed that the genes are located in the euchromatic region because that they are active, transcriptionally they are active, and in the heterochromatic region there are many other sequences which are not active transcriptionally. And on the right of it, you will see the chromosomes which are at a level which is called as pro-metaphase level. So we have tried to map the hetero and euchromatic and also the repetitive DNA using fish technique. Again, this is another advancement which was made, and we have tried this technique in case of Plantago ovata. Again, we have tried to map the histone modifications. So we have, for example, you can see on the left DAPI, this is the normal stain, and then we have used the antibodies, and you can see on the right the merger, wherein we are trying to merge the these are called as epigenetic marks. This is another way of finding out which regions are active in chromosomes, and what you can see on the extreme right, that is the merged one, is possible because of the in situ hybridization. So you can localize the antibodies or histone antibodies, you can localize and try to determine which areas of the chromosomes are active so far as they are gene rich or they are gene deficient. Now here I just wanted to tell you that earlier I said that the plant chromosomes, they have telomeres at the end. You can see them glowing at the ends, and here we have used Arabidopsis sequence for finding out the telomeric sequences in ovata, telling you that the telomeric sequences resemble in Arabidopsis as well as plant type of ovatas. These are the ends which are preventing the chromosome from circularization. Now, a very important thing which happened, as I said, in 50s, even up to late 80s or 90s, even today people are trying to develop the maps. And there are three kinds of maps which you may have heard of. One is the genetic map. So how do you locate genes on the chromosomes? One of the ways is genetic map, for which you need different alleles of a marker are required, and based on percentage of recombination between markers, the markers could be any RFLP marker, AFLP marker or SSR markers. They can be then based on percentage of recombination between markers, they can be measured in centimorgans, and then we report a linear order of markers and the amount of recombination between linked markers. That is how a map is developed. But these kinds of maps, the genetic map, not provide any information on physical distance. How far are they physically? That is possible with genetic maps. And genetic maps, as I said earlier, they can be created with RFLP and AFLP markers. Then comes the cytological maps. As I told you, position of microscopic structures on chromosomes. There are very few, like we have the centromere, we have the heterochromatin, we have NOR, nucleolus organizing region, we have the euchromatic regions, there can be knobs as are true in base, chromomeres, and later when other landmarks were added like banding, with C banding, R and Q banding, and with fish. So these are, you can develop a cytological map. So what is basically a map? You have linearly to show a chromosome and you have to add to that is markers. But you can understand that there are very few markers and there are limitations. Then comes the cytogenetic map. That is where we combine both the genetic map as well as the cytological map. So we use cytological and genetic position of markers simultaneously. Cytogenetic map will show where recombination is likely to occur. That is identification of gene-rich regions. As I said, if you are able to map the euchromatic and heterochromatic regions, one can say that where exactly our gene is located, so you can develop such maps accordingly. Now then, maps are integrated by locating markers relative to breakpoints, like you can identify cytologically the deficiencies, duplications, inversions, translocations, and position of breakpoint on genetic map can be correlated with physical position of breakpoint in cytological location. So in a way, if I put it very simply, if you have a genetic marker, an RFLP marker, and if you have a cytological marker, say a heterochromatic region or a centromeric region, so both can be combined to develop a map which will be a physical map. So you will exactly know that how far is this marker from the centromere or how far is this marker from the telomere. So this gives you a better perception about the position of these markers on the chromosomes. Let me tell you that scientists have used these techniques extensively and they have got greatly benefited by using these cytological and cytogenetic maps. Now, if you see the history of this mapping, it was Barbara McClintock in 1931. She is credited with the initiation of cytogenetic mapping in plants. Barbara McClintock is the famous scientist who has been awarded Nobel Prize for her work on jumping genes, as we know, transposable elements in base. She was the first to identify unambiguously the 10 packetine bivalents of genes. Packetine is a stage in meiosis. In the interphase stage, you have then the packetine, diploidine, diakinesis, and so on. But this is one of the stages when the chromosome length is very good and you can easily identify all the chromosomes at this particular stage. So she could locate few genes, like for example, colored aileron, Duncan endosperm, and maxi endosperm, just 3 genes. But as you know, there are hundreds and thousands of genes in a particular genome. So if you really want to map them, then you need other techniques. Now, several challenges were faced, and because of this problem, limitation of the technique, that it could not be more genes could not be mapped. Then came Casperson in 1969. He came up with a technique which is known as Q banding. That is, quinacrine fluorescent dye was used to band the chromosomes. Band would mean that there were specific regions, they could be identified as quinacrine positive and quinacrine negative bands in the chromosome. Pardue and Gall, 1969, developed GeneSA C banding. I will show you a photograph here. Yeah, you can see it here. See, this is the typical C banding, as you can see. This photograph is derived from Figueroa and co-workers paper of 2012. Now you can see these dark spots. They actually go to the heterochromatin regions, but with this becomes a sort of a fingerprint for a chromosome. So a typical chromosome can be identified by using the pattern. It will always be the same pattern or the same chromosome. I'll explain why I have put this photograph here. In wheat, a novel system of gametocidal genes was used to produce stable chromosome deletion lines. Now this is a chromosome of wheat, 5b. These are all 5b chromosomes. But you can see that gradually they are missing some region. Right from here you can see this terminal part is missing, so it has been deleted. There is a gene located in wheat, in some accessions of wheat, which lead to deletion of these segments. Now how to detect that how much of the part has been deleted? It was with this technique. And you can see here this band, gradually if you see the other chromosome, they miss this last chromosome. It does not have anything of these bands here. It became possible because of this C banding technique. One could easily identify which regions have been lost. Now these deletion lines are even used today. Ando and Gill, 1996, they used this GC system in conjunction with C banding to produce and characterize chromosome deletion stocks of common wheat. About 400 deletion stocks have been identified and they are being used throughout the world. These are very, very important stocks, genetic stocks, which have been produced because of this cytogenetic research, and they have been used for mapping now. Now, the best part was that with in situ hybridization, this mapping became very, very easy. The labeled DNA or RNA sequences are used as probes to hybridize with complementary sequences in target DNA. As I told you earlier, radioactive tritium or iodine labeled probes were used for mapping knob repeats of maize way back in 1981, and ribosomal DNA genes of rice in 87. In 1985, Rayburn and Gill, they combined iodine tritium. Since one mapping has been undertaken in numerous recently in 2008, used nanocrystals called as you know, lagging based fishy and presenting search need for map. Now the markers which have been used for are, you can have an RF and use it to fish as a probe, LP, SSR. You can use the DNA track. The yeast chromosome labeled mapping maps of when you are labeling in and things are genomics. So today we are the studies which have been conducted as many of our uh, my own in other uh, uh, this chromosomes extend it first all these targets in a not for with you about special types. I have been associated lab associated since last several years. That is, I will start something I have done with me and uh, was in 1907 per new chromosome. They first noticed in rye and maize. They this them from chromosomes. So complement a plant is as complement is B chromosome first used all in 20 was working. Male and B and two pseudophyte and fungi, and their properties have been very well documented. Essential features of these are that they are dispensing, combine A chromosome during inheritance. Create problems, especially for there are now studies have been so the chromosome major it contribute has what all in the recent past and they want to have useful attributes in planting. They are dispensable, they may be present in and advantage issues they cannot represent, and the most important characteristic that they do not recombine with A's during meiosis. So there is no exchange of genes, there is no recombination between A chromosomes, that is the normal set of chromosomes, and the B chromosomes. Now let us discuss first rye as a model, then I will come to my work or our work, sorry. And rye really can be discussed as a model because a lot of work has been done in rye B chromosomes. Relative sequence analysis of A and B chromosomes of rye repeat in unexpected results that these RNA fragments derived from multiple A chromosomes, multi-chromosomal origin of B chromosome. Earlier they were thought that B chromosome arises from the A chromosome, but now yes, it arises from A, but it has a multi-chromosomal origin. Rye Bs were found to have a large amount of B-specific repeats, that is the repeats which are not found in other A chromosomes. and had insertions of cytoplasmic organism. They would have DNA from the that it seems that B acts like a genomic sponge which in and maintain the sequences of is actually now organelle DNA. You can have that is one therefore, but the sequence analysis and composition and distribution of high copy sequences in the IP that B's contain a similar proportion of repeats as in the A chromosome, but differ substantially in repeat composition. Now, if you see the most abundant mobile elements, gypsia, in the URI are similarly distributed along A's and B's, but an ancient retro element, Sabrina, is less abundant on B's than on A. In contrast, active element revolver as well as predicted copia retrotransposon disproportionately abundant on. Thing is going on between the during the evolution between A chromosomes and B chromosomes. Some of the repeats they become B specific, and some are the ones which are equally distributed or similarly distributed between A. The functional significance remains to be seen. So far as this B chromosome is C chromosome, and that's why they are mechanism that early that is located in the but it would one has two families. There are some special group that high copy sequences distinct evolution for IB very clearly that this B chromosome currently it has mitochondrial DNA, it has chloroplast DNA, it has many other repetitive sequences which are the specific to B chromosomes, and you can see these last ones here. They are the B specific non-disjunction non-control region. This is the region which I was talking about. If you remove not shown on this stated in the next generation. Exam survival. Now, why are these chromosome in every it you know is not coding. Coding or that while elements and other sequences in the interesting thing that still many people say that it is still an enigma that how did these group they have come out with a step model for the origin of B chromosome. They say that first a proto B chromosome is formed by segmental or whole genome duplication. Integrated and being in the chromosome model gravel which has been proposed by Huber. I will tell you that we have also proposed a model way back in 2002 and I will show you that that is perhaps the best acceptable model so far. Now I will come to B chromosome in our plant for which we have worked in our lab here. I started my PhD program on this particular problem. That was at that time we were not sure that what is the nature of this particular chromosome. So this chromosome at that time we called as microchromosome. It was present in addition to the normal complement of 12 chromosomes. The chromosome was later when we did all kinds of transmission analysis and other studies, meiotic studies during my PhD. But later we found that chromosome is completely heterochromatic when we use the C banding analysis. Now this B chromosome is unique. For a very long time we were not naming it as B chromosome. We were still not sure that it is a B, but later when we saw the characteristics of this chromosome, we decided to call it a B chromosome. It is unique in having its entire body mass derived from 5S DNA sequences. We were the first to present the experimental evidence of de novo origin of novel B chromosome in Plantago through specific DNA sequence amplification. I will elaborate on this point later. Using fish and reverse Gish techniques, the entire chromosome was found to get painted with 5S rDNA probe. When this particular study was conducted in the year 1999, I was at Kansas State University. It was the most thrilling, I would say, event of my life when I found that is getting completely painted with the 5S rDNA probe, telling me that it has at the ends the 45S rDNA, I will show you, and telomeric sequences, and rest of it is completely filled with 5S rDNA probe. Believe me, this was the first report wherein a chromosome was found to be completely made up of 5S rDNA. Now this is the plant. This is the plant of Plantago legopus, which has 12 standard chromosomes and 1B was present here, and in this plant we had 12 plus 2B. And when we trace the origin of this particular plant, we found in the year 1988, I found in some cytological preparations chromosomes like this, ring shaped. So this only told me that these chromosomes have lost the telomeres. That is why they have become rings. And you can see them at metaphase as well, big rings. And later in the progeny of this plant, we found a plant which had these 12 standard chromosomes and one chromosome which was completely heterochromatic on C banding. This had another plant. In its progeny, we got a plant which had now two such chromosomes. So you can see a plant with 12 plus 2p. This work has been published in Chromosoma, which is one of the I would say the topmost journals so far as chromosome research is concerned. And here comes the photograph. I told you earlier that this is the one which you see in green here, fluorescent labeled chromosome, which is here in the green, tells you these are the normal. On other chromosomes, these are the 5s ribosomal RNA sites, and you can clearly see that this chromosome is completely painted with 5s ribosomal RNA. When we use the same cell with other probes, and you could see the 45s, the ones in the red signals which you can see at the end. So these are the 45s RNA and ribosomal DNA sequences, and this is the one cell having two such P chromosomes. Now you can see on the right the sketch, the schematic I have shown, that the entire chromosome has 5s rDNA. There is a centromere, there are telomeres at the end, then followed by the 45s DNA. This was a very exciting work which we published in Chromosoma, and later by using the fluorescence in situ hybridization, we found that there are some additional 5s sites also in some chromosomes, and with telomere, I told you that we used Arabidopsis telomeres to find out, yes, it has this P chromosome also has the telomeric sequences. Now subsequently, my research team, the students, PhD student Gurmeet Kaur, she studied this further, and in the year 2017 we published this work wherein we showed that we could have a plant wherein which the chromosome number was 23, and when we used fluorescence in situ hybridization, we were surprised to see that there are 5 P chromosomes. All of them are getting painted with the 5s ribosomal DNA probe, and we also had a plant with 28 chromosomes, but unfortunately we couldn't use that for fish, so we are not sure, maybe it has more than 5 P chromosomes in its genome. Now we also did fish on the meiotic chromosomes, and so you can see at metaphase, this is the meiotic metaphase, and you can see these chromosomes can be easily detected by using the 5s rDNA as a probe. Now this P chromosome in Plantago lagopus is cytogenetically very good technique for and co-workers in our lab. We further characterized this chromosome and report mixture of rDNA sequences and transposable elements. Initially, we are always thinking that it only has the 5s rDNA, but when we use some transposable sequences, also transposable elements, they were also mapped on these chromosomes, on this chromosome. Then recently, what the DNA composition of Plantago lagopus, next generation sequencing, we have sequenced the repetitive fraction of Plantago with and without P chromosomes, and we have in silico characterized using advanced sequencing technology with highly and moderate layer genome was found to be relative repetitive sequencing, making up to 68% of the genome. A centromere specific marker and a B specific satellite and repeat enriched in polymorphic A chromosome segments were identified. We identified a B specific that found Ullmann's amplification, including 5s rDNA fragments and other repetitive DNA elements. Now here is the picture of the fiberfish, where we tried to map these 5s, and we could easily identify that there are gaps. You can see it here that there are gaps in the 5s. This is a continuous DNA fiber which has been labeled or hybridized with 5s rDNA, and you can easily see the gap. The nature of this gap tells us that besides 5s, that is why I said when we use the repetitive element, the PY1 copia element, we found that was also getting mapped on particular chromosome. The conclusion in 2013 in Cytogenetics recent paper on this aspect on cytogenetic and genome research using these probes which we got from the next generation sequencing. When we use from them these probes on with fish, and we found that you can clearly see even at packeting. This is a packeting stage of Plantago and you can see these two chromosomes. This is a cell having two B. It has this PL SAT B. Can easily you can see these bands here. They are all of P SAT B. Even at beta phase, you can't see them with such clarity because beta phase is a highly contracted chromosome. You can see it here. And similarly, we use other repetitive sequences which were specifically isolated because of our NGS data, and we developed the probes and we could now know as Plantago Lagopus. Here we even analyze LSAT, which are getting all throughout the, but basically this PLSAT has other sequences as well, but 5S and 5S ribosomal RNA is there. There we have made the alignment which are similar, right? And initially when we 5S, very sure that what we are getting from are not only 5S RDNA but also several of these repeats. You can see here repeat clusters which we have done the analysis of NGS with CL4 or CL94. These are the repetitive fractions which are enriched in this P chromosome. Paper published in 2016. Now this is the picture which has finally emerged. Last year in 2019 we published this paper showing that this was the first one, is the initial which I showed you, our 2002 paper. So here we had P80 that applied, then we had the 45S that was determined by PA71 probe, then we had PPOV1 probe that was for 5S RDNA. So you can see that the same picture, telomeres followed by 45S followed by 5S. Later, as I told you, we isolated some transposable elements them, so those were mixed intermixed with this 5S, and finally the picture which has come through next generation sequencing, we are now giving you picture chromosome not only contains the also intermingled this, and interestingly found organelle DNA in the position. So this chromosome has evolved so along with one or fragment chromosome its own